I'm an aquatic ecologist. I'm interested in studying algae blooms and algae growth at Chautauqua Lake and how that relates to water quality issues. So we study a lot of different aspects of algae growth. We're looking at the species that are present. We're looking at the water chemistry and quality, water clarity. Right now we're looking at some other features like temperature within the lake, nutrients, all the factors that contribute to algae growth. Right now we're working on understanding some of the bigger spatial patterns at Chautauqua Lake in terms of the temperature layers within the system, how that might change when a storm event comes through, what that might mean for nutrients moving within the lake itself, from the sediments up to the water and back down again. And so we're still in the early stages of that as we're collecting those data.
The story for us really starts before we get on the boat, because in the work that we're doing, we're really selective upon where we're interested in doing our research. Based on the bathymetric map, there is a deeper hole just off the bell tower on Chautauqua. And so by launching off the shores at Chautauqua, we're able to locate that deeper depth. And then one of the first things we do in our notebook is we take field condition notes. And then usually the first thing that we do is we take a vertical profile. And so a vertical profile, we use a Hydrolab multi-probe sonde. It will tell us the depth at which the sonde is at. It will tell us temperature, pH, conductivity, dissolved oxygen, and chlorophyll. And what we do is we start at 0 meters right at the surface, and then we drop down every meter or about every 3 feet until we reach the sediments. So we go ahead and wherever that big chlorophyll spike is based on our vertical profile, we take our first Van Dorn sample. So the Van Dorn, we drop it down into the water, and then we pull it up and we're able to collect a water sample from 3 meters in the water column. The one thing that we do is we take those Van Dorn samples that we had, that we took at the known depths, and we take those samples and we process those back at the lab. One of the things we do is we use an acid washed bottle, and we process that for nutrients. In Chautauqua, we're specifically looking just for residual phosphorus and total phosphorus. We also take out of our soap wash bottles, we'll look at chlorophyll, and we'll filter out for chlorophyll at each depth just to get the overall algae biomass reading. We will eventually process both the nutrient and the chlorophyll sometime later on in the season. We do chlorophyll about every 2 weeks, but nutrients we will save just for the end of the season.
The appeal of working at Chautauqua Lake is being able to interface with the community a lot. So as a scientist, sometimes our work feels kind of far away from the people it might affect. And in this case, it's really been wonderful to have the opportunity to do outreach and to connect with community groups and listen to the people who live on the lake and who are impacted by the water quality. And it really brings the science home for me. It's easier for me to see how people relate to their lake when I'm bringing my science and I get to talk about that with them. And then it's a fun place to work. It's never boring.